Hello, I'm Fred Bookstein. I would like to introduce you to several new tools of visualization for the measurement of biological shape. These techniques are all ultimately aimed at the description of change or variability in images that are calibrated by biological landmarks, points that have the same names from specimen to specimen, like the bridge of the nose or the tip of the chin. Examples from inside the brain will be introduced near the end of this string of demonstrations. The techniques I will show you here include the use of thin plate splines for measuring the linear and nonlinear parts of arbitrary shape changes, the application of spline-based warping techniques to two-dimensional images, the averaging of unwarped pictures, and the use of the splines in 3 dimensions to support digitizing and understanding solid medical images. The two-dimensional examples have been computed in X windows. The 3-dimensional rendition of the principal warps is drawn using AVS, Stardon's visualization system, and the sketch of morphometrics for solid brains is programmed using calls to the 3-dimensional graphics interface XFDI. The first demonstration will show interpolation between arbitrary sets of matching points by a thin plate spline. The spectrum of bending energy of that spline can be used for a very useful decomposition of the warping into features in a hierarchy of physical scales. Begin with the simplest case, 3 points in a plane that are matched to 3 others. Let's visualize this shape change by its effect on a square grid. If the 2-point sets are identical, the grid is unchanged. As we move any of the points in the right-hand set, the grid changes to correspond to the uniform transformation implied. We begin the feature extraction by copying this linear part to the bottom left of the screen, where it will become one in a series of components, partial warps, pertaining to maps of any number of landmarks. Add a 4th point to this little scheme. Now the transformation is no longer uniform. Still, as any corner of the square is displaced, there is induced a smooth interpolation, which is broken down in the lower row into a pair of components. The one at the left is still affine, the transformation of square into parallelogram. What is the component at the right? The interpolation we are using here is the thin plate spline. This is the exact interpolant among these pairs of 4 points for which the integral of the total squared quadratic variation, the sum of all the squared second-order partial derivatives, is a minimum. The integral is taken over an infinite screen. For two-dimensional data, this interpolant is a linear part added to a sum of vector multiples of the single function r squared log r. But the pattern of those multiples is controlled more rigorously than that. All the mappings we saw differed from some linear transformation by a vector multiple of this one single surface, the only principal warp of a starting square of landmarks. This surface corresponds to the form of a thin metal sheet lifted a certain distance upward at the ends of one diagonal of the square and dropped the same distance at the ends of the other diagonal. As you can see here, it has a lot of symmetry. Rotating it by 90 degrees is the same as turning it upside down, and so on. Back in the plane of the data, we can now see how this single sheet permits us to express any shape change of the starting square. Moving this corner right, for instance, generates an affine part at the lower left, together with a horizontal multiple of that one sheet at lower right. Upper left and lower right corners move right, and the other 2 move left. Moving the corner along a diagonal, another view of the same sheet. Moving the corner a long way in that direction almost annihilates one component of the affine part. Let's add a little more complexity to the picture. One additional landmark point in the middle of the square. In the lower row, we now see a decomposition of the interpolated smooth mapping into an affine term together with 2 gridded features. What are these 2 other terms? The features at the bottom, the partial warps of this 5 landmark transformation, are combinations of 2 principal warps, principal modes of bending. These are eigenfunctions of the matrix of bending energy underlying the formula for the thin plate spline as a matrix transformation of the coordinates of the landmarks on the right. This is the principal warp of highest specific bending per unit landmark displacements. You see that it represents the contrast between the displacement of the middle landmark and the displacement of all 4 of the others. This is the feature of the warping that has the smallest possible geometrical scale. Away from the vicinity of the landmark data, it flattens out. So did the 4-point warp in the previous sequence. Besides the affine part and this high-energy principal warp, there is one more component of the change in shape of that quincunx. It is the same as the thin plate for the transformation of the square of outer landmarks that we already saw. Transformations described by this component move the middle landmark precisely in accord with what the outer corners are already doing. That's why in this picture that middle landmark lies on the sheet precisely in accord with the larger scale bending specified by the corners. Back in the space of our data, we can see how these components combine to measure particular shape changes. If we move only the center landmark, the resulting mapping is dominated by the component of change corresponding to the first, the sharper principal warp. If we move a corner of the square, the dominant component is the one for the 4-point, 2-up, 2-down warp we saw earlier. There's also a small contribution from the sharper partial warp representing the relative displacement of the middle landmark. That's because the middle landmark, even though it didn't move, is no longer located where the map based on the other landmarks would put it down. If we adjust its position to correspond exactly to where the 4-point interpolation would set it down, then the sharper partial warp goes away entirely. When landmarks don't start with so much symmetry, usually the feature of their interpolant having smallest geometrical scale takes the form of a contrast among the landmarks that are closest together. Here we show a large triangle of landmarks with two others juxtaposed in the interior. In this case, the thin plate corresponding to the sharper of the principal warps looks very much like the potential map of a dipole you may have seen in a physics course. The surface is sharply crimped between that pair of interior landmarks, raised high at one, dropped at the other. This stands for the discrepancy between their separate displacements after controlling for the uniform part of the shape change, a part specified mostly by the triple of landmarks set out at the corners. In between this feature of the shape change and the affine part is the curving feature of largest geometric scale. In this case, that takes the form of the bulge we saw a moment ago. This feature stands for the displacement of the landmarks inside the triangle considered as a pair with respect to those located at the outer corners. This decomposition of features applies to the thin plate spline interpolant on any number of landmarks in any configuration. Any transformation decomposes into an affine part together with a sum of multiples of a series of principal warps representing steadily higher amounts of bending per unit landmark displacement. Back in the plane of the T of landmarks, we can see how this decomposition applies to actual rearrangements. If I move the upper central landmark to the left, I have induced a feature which is entirely horizontal and entirely restricted to that sharpest warp, the one whose surface looked like a dipole. If instead I move both of the central landmarks downward, the induced transformation has no dipole component, but a sizable admixture of the largest scale principal warp, the contrast of the middle two landmarks against the outer three. If I then restore the horizontal glitch to the upper central landmark, there results a map that has components of bending at both geometrical scales. A horizontal multiple of the sharper dipole principal warp, which you see in the partial warp at lower center, and a vertical multiple of the broader principal warp seen as the drop of the whole center of the grid in the other partial warp at lower right. In these demonstrations, the landmark locations have been whatever I said they were. Those green dots landed wherever they pleased. In applications to real biomedical images, they'd better represent data. Here's a brief introduction to the use of the splines for specification of landmarks. We have here a sketchy representation of the outline of the shape of the mandible in a stereotyped pair of cotton rats. Perhaps these represent specimens in two habitats. We'd like to describe the relation between these two shapes by drawing a thin plate interpolant from one to the other and then extracting the features of that thin plate spline. So we need some landmarks to represent the information underlying the measurement. Let's play with the location of that last landmark a bit. Here we have a spaced-out cotton rat jaw, here a Neanderthal. We aren't going to make very much progress this way. Nevertheless, if this set of three landmarks had been sufficient to superimpose the full outlines, then the comparison of forms would have been borne by this transformation grid and would be measured by its features, just as in the preceding demonstration. Let's proceed to add landmarks, one on the condyle, one at the chin. We're doing a little better. We can continue with a pair of landmarks at the front and back of the tooth row, and this is a good place to stop, though we could continue the matching as far as we liked. The larger scale features of the transformation are already encoded. These include a transformation that is mostly size adjustment without any change of shape, here in the middle of the ramus. The grid cells are not too far from square. And near the teeth, a strong local reduction in scale together with a strong shear. We could continue by decomposing this general change into components just as in the previous part of this demonstration, but this particular change can be eyeballed. Apart from the linear term, the transformation is mainly a multiple of the largest scale principal warp, the 2-up, 2-down sheet we saw first. The preceding computations go in real time on any high-speed workstation. I turn now to a computation that requires about half a minute as presently coded, warping entire 256 squared grayscale images. Here's a mid-sagittal section of one clinically normal human brain as imaged by MRI. The green dots are the locations of 13 landmark points that I will name for you later. We can use these landmark locations to drive a warping of the entire picture as follows. Suppose a warp is specified from the left-hand configuration to another. The warped picture is computed by running the warping function backward from the right-hand landmarks back to the original set, then copying or interpolating pixels. This is a fairly slow procedure, so we demonstrate using Steve Peiser's trick, filling in successively less informative edges down to 5% of the total. When the mapping is satisfactory, here we've turned a medical student's brain into Bart Simpson's, we go back and specify that all pixels should be transformed, not just the 5% that are most edgy. The tape cuts about 30 seconds of CPU time here to show you the resulting image. For that demonstration, the picture to which the medical student was warped was arbitrary. In practice, we use exactly the same procedure to construct averaged images by registration upon arbitrary sets of landmarks. John Mazziata, Dan Valentino, and I took 14 mid-sagittal sections from UCLA medical students who were otherwise normal and averaged the locations of these 13 landmarks using the statistics of landmark locations I've been developing over the years. Every picture was then warped to this average. Remember, that means the average was warped into every picture and the pixels transcribed. Then the copied pixels were averaged one at a time. So you're looking at a rigorous average of 14 sets of gray levels in the same geometry. I will show you some of the consequences of this strategy for the look of the average picture, but first let me introduce you to the landmarks we used. The front of the frontal lobe, the genu of the corpus callosum, anterior commissure, optic chiasm, top of the pons at the peduncles, the bottom of the pons at the medulla, obex, tip of fourth ventricle, inferior colliculus, posterior commissure, the splenium of the corpus callosum, the intersection of the parieto-occipital and calcarine sulci, and the back of the cerebellum. These are of different reliability and homology from case to case, of course. The statistics of those measurement issues is an open research topic. The details of picture averaging may be viewed better in enlargement. This is not conventional stereotaxy, a matter of correcting for horizontal and vertical proportions. There's a considerable component of vertical unbending here as well. In the midbrain, the landmark registration is really astonishingly good for edges. Sharp contours emerge, like the front of the pons and the shape of the fourth ventricle, corresponding to arcs tacked down only at their endpoints. In this region, indeed, the young doctors seem to all be built on the same principles. Once the landmark points are in place, the organ boundaries follow. The corpus callosum, locus of connections between the two halves of the cortex, is an interesting intermediate case. The curve of its arch is detectable in the averaged image, but it is not terribly clear. There is still individual variation remaining, variation whose correlation with mental properties might prove very interesting. At the back of the vault, we find another failure of registration owing to lack of appropriate landmarks. One case soars upwards out of the picture. At the forehead, likewise, we have no landmarks. Hence, the outer and inner skull tables do not line up very well. As with the embouchure of the cotton rat, we could certainly improve this fit with additional data, and then inquire as to whether the features of the cortex could be made to align. Below the frontal lobe, in the maxilla, of course the superposition is noise. Extrapolation to non-neural structures outside the brain case is not possible even in principle. This single averaged picture together with the statistics of the standardization to this shape, may be sufficient for functional studies throughout the midbrain. I have one more morphometric tool to show you, a prototype for the extension of all the preceding computations to solid medical images in 3 dimensions. The following uses a single solid MRI scan sent to me by John Mazziata of UCLA. Interaction with this program uses 2 windows. The larger one on the left will show variously a slide of the student brain, a wire mesh model of a deformed piece of fruit, or both. The smaller window on the right has 2 panels. The upper circle represents a sort of nonlinear trackball. The mouse will click in there to specify rotations of the north pole of the point of view. The lower part of this panel, the square, is for specifying displacements of selected landmarks in the plane of the current projection. These are sections oriented mid-sagittally, vertically up the middle of the head. We can march back and forth across the head in the plane of the screen, or rotate the slice just enough to show that these are resampled slices of a solid in 3 dimensions. For now, ignore the colored polygons. They will be explained in due time. Changing the plane of section by 90 degrees brings us around to the horizontal planes you may find more familiar. We can march these down through the medulla and the ears, or march them up through the lateral ventricles and the upper structures of the cortex until we run out of slices in this particular data set. In addition to this particular brain, there is another graphical object in this window, a representation of part of the standard or average brain. We don't have any data for this structure yet, so I'm using a standard grapefruit instead, with its suspiciously smooth faces colored arbitrarily. This grapefruit sector is delimited at present by 4 landmarks shown here by the red spheres. 4 landmarks in 3 dimensions specify an affine, linear transformation, just as 3 landmarks did in 2 dimensions. We can grab a landmark, it turns into a spray of red lines, and drag it to another location. There results a shear of the entire grapefruit sector. Again, just as in 2 dimensions, an exact interpolant for any number of landmarks is supplied by the thin plate spline. In 3 dimensions, the basis functions past the affine part are absolute value of r, rather than r squared log r. There is otherwise no difference. The thin plate interpolant is still the least bent mapping consistent with the landmarks supplied. It can be relaxed if the landmarks are known to be noisy, but I will not show this here. As we add new landmarks, then drag them around, the grapefruit is smoothly deformed into a shape that could be an interesting biological object, such as a part of the very brain we are looking at. With support from the NIH, we are building software for matching an averaged brain to specimen patient images by iterative deformation, the same technique you saw applied in 2 dimensions for the mandible of the cotton rat. The general idea is to tack down enough landmarks that the deviation between one image and the warping of the other is not worth pursuing further. For this little prototype demonstration, let's match our grapefruit sector to half the top of this student's skull. We begin with a horizontal section through the anterior commissure. The grapefruit sector has 3 landmarks in this plane. This one must be sent to the posterior pole, this one to the anterior pole, and the picture cleaned up a bit. There is one more landmark in this plane which needs to be manipulated so that the curving lateral outline of the patient's skull can match that of the standard. To do this, we first move the landmark in a bit and then adjust it a little upward to take account of the slight bulge of the back of the calva with respect to the front. There is one more landmark on the grapefruit, lying in the mid-sagittal plane at the very top of the skull. To align it, we restore the original mid-sagittal plane of section, only to notice that the landmark is outside the picture we were originally supplied. No problem. We can select it and drag it around anyway, until the contours of the grapefruit generally match those of the skull in this plane as well. At this point, we've finished the digitizing of this patient's skull vault, except to verify that the deformation-based match given by the thin plate spline is close enough as far as this structure is concerned. To this end, we play with many alternate views, any of which could reveal discrepancies between the 2 surfaces if their sections didn't line up. We can show the section cutting through the grapefruit in any of a variety of orientations, or we can erase the grapefruit mesh and show only its locus of intersection upon the actual plane of the picture. That's the colored lines you've probably been noticing when the grapefruit surface wasn't being shown. If the digitization is adequate, then whatever the plane of section, the boundary of the curving part of the grapefruit will line up with a comparable white line of bone. we can find no orientation or plane of section in which the grapefruit and the case are seriously out of alignment. This completes the demonstrations I wanted to show you. I began with powerful decompositions of changes in any configuration of landmarks. I showed you how the interpolant underlying this decomposition, the thin plate spline, could be used to compare outlines of two-dimensional forms. I demonstrated applications of this technique to picture averaging, with clear implications for subsequent statistics, and I showed you the prototype of the system for carrying out the same cycle of mapping and matching in three-dimensional images. The key to all these visualization tools is the use of the thin plate spline in a dual role, as a technology for interpolation and as an algebraic means for decomposing sets of landmarks into components corresponding to the eigenfunctions of that interpolation. The combination of the thin plate algebra with the visualization tools of a modern high-speed workstation makes a very promising platform for investigations into the statistics of normal and abnormal form and for correlating image properties with biological predictions or classifications. Thank you.